Hello students, you know proteins are the most scoring topic in class 12 biomolecules. Every year you get at least one question from denaturation and structure in CBSE, NEET and other state boards. I am going to break down NCERT chapter Biomolecules in few minutes. No fluff. Will cover classification, structure, labels, denaturation with example and the exact lines NCERT. Accept your answer. Let's start with what a protein actually is. Proteins are the most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. Proteins occur in every part of the body and form the fundamental basis of structure and functions of life. Proteins are also required for growth and maintenance of body. The word protein is derived from Greek word proteos, which means primary or of prime importance. All proteins are polymers of alpha amino acids. Amino acids contain amino single bond NH2 and carboxyl single bond COOH functional groups. Depending upon the relative position of amino group with respect to carboxyl group, the amino acids can be classified as alpha, beta, gamma, delta, and so on. Only alpha amino acids are obtained on hydrolysis of proteins. They may contain other functional groups also. All alpha amino acids have trivial names which usually reflect the property of that compound or its source. Glycine is so named since it has sweet taste. In Greek, glykos means sweet, and tyrosine was first obtained from cheese. In Greek, tyros means cheese. Amino acids are generally represented by a three-letter symbol. Sometimes one letter symbol is also used. Structures of some commonly occurring amino acids along with their three letter and one letter symbol are given in table. Amino acids are classified as acidic, basic, or neutral depending upon the relative number of amino and carboxylic groups in their molecule. Equal number of amino and carboxylic groups makes it neutral. More number of amino than carboxyl groups makes it basic, and more carboxylic groups as compared to amino groups makes it acidic. Essential amino acids and non-essential amino acids. Out of the total 20 amino acids found in nature, 10 have been classified as essential and the remaining 10 as non-essential. Non-essential amino acids. The amino acids which can be synthesized in the body are known as non-essential amino acids. Essential amino acids. Amino acids which cannot be synthesized in the body and must be obtained through diet are known as essential amino acids. Amino acids are usually colorless crystalline solids. Amino acids are water soluble, high melting solids and behave like salts rather than simple amines or carboxylic acids. Note, salts like behavior of amino acid is due to the presence of both acidic carboxylic group and basic amino group groups in the same molecule. Jitter ion. In aqueous solution, the carboxyl group can lose a proton and amino group can accept a proton, giving rise to a dipolar ion known as Jeter ion. This is neutral but contains both positive and negative charges. Amphoteric behavior of amino acids. In Jeter ionic form, amino acids show amphoteric behavior as they react both with acids and bases. Optical activity. Except glycine, all other naturally occurring alpha amino acids are optically active since the a carbon atom is asymmetric. These exist both in D and L forms. Most naturally occurring amino acids have L configuration. L amino acids are represented by writing the NH2 group on left hand side. Structure of proteins. Proteins are the polymers of alpha amino acids and they are connected to each other by peptide bond or peptide linkage. Peptide linkage. Chemically, peptide linkage is an amide bond formed between COOH group and NH2 group dipeptide. The reaction between two molecules of similar or different amino acids proceeds through the combination of the amino group of one molecule with the carboxyl group of the other. This results in the elimination of a water molecule and formed a peptide bond, single bond CO, single bond NH, single bond. The product of the reaction is called a dipeptide because it is made up of two amino acids. Example, when carboxyl group of glycine combines with the amino group of alanine, we get a dipeptide glycyl alanine tripeptide. If a third amino acid combines to a dipeptide, the product is called a tripeptide. A tripeptide contains three amino acids linked by two peptide linkages: tetrapeptide, pentapeptide, or hexapeptide. When 4, 5, or 6 amino acids are linked, the respective products are known as tetrapeptide, pentapeptide, or hexapeptide, respectively. Polypeptides. When the number of such amino acids is more than 10, then the products are called polypeptides. Protein. A polypeptide with more than 100 amino acids residues having molecular mass higher than 10,000 units is called a protein. The distinction between a polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids are likely to be called proteins if they orderly have a well-defined conformation of protein, such as insulin, which contains 51 amino acids. Classification of proteins. Proteins can be classified into two types on the basis of their molecular shape. One, fibrous proteins, and second, globular proteins. Fibrous proteins. When the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. Some common examples are keratin, present in hair, wool, silk, and myosin, present in muscles, etc. Globular proteins. This structure results when the chains of polypeptides coil around to give a spherical shape. These are usually soluble in water. Insulin and albumin are the common examples of globular proteins. Structure and shape of proteins. Structure and shape of proteins can be studied at four different levels, that is primary structure, secondary structure, tertiary structure, and quaternary structure. Each level being more complex than the previous one. Primary structure of proteins. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence. And it is this sequence of amino acids that is said to be the primary structure of that protein. Any change in this primary structure, that is the sequence of amino acids creates a different protein. Secondary structure of proteins. The secondary structure of proteins refers to the shape in which a long polypeptide chain exists. They are found to exist in two different types of structures, such as alpha helix and beta platelet sheet structure. These structures arise due to the regular folding of the backbone of the polypeptide chain due to hydrogen bonding between single bond CO single bond and single bond NH single bond groups of the peptide bond alpha helix structure. Alpha helix is one of the most common ways in which a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw, that is helix, with the NH group of each amino acid residue hydrogen bonded to the CO of an adjacent turn of the helix as shown in figure beta platelet sheet structure. In beta platelet sheet structure, all peptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonds. The structure resembles the plaited folds of drapery and therefore is known as beta plaited sheet structure. Tertiary structure of proteins. The tertiary structure of proteins represents overall folding of one polypeptide chains, that is further folding of secondary structure. It gives rise to major molecular shapes with fibrous and globular. Note the main forces which stabilize the secondary and tertiary structure of proteins are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic force of attraction. Quaternary structure of proteins. Some of the proteins are composed of two or more polypeptide chains, referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. Protein found in a biological system with a unique three-dimensional structure and biological activity is called a native protein. When a protein in its native form is subjected to physical change like change in temperature or chemical change like pH, the hydrogen bonds are disturbed. Due to disturbed hydrogen bonds, globules unfold and helix get uncoiled and protein loses its biological activity. This is called denaturation of protein. During denaturation, secondary and tertiary structures are destroyed, but primary structure remains intact. The coagulation of egg white on boiling is a common example of denaturation. Another example is curdling of milk, which is caused due to the formation of lactic acid by the bacteria present in milk. That's proteins done from NCERT. Remember the four structures and the difference between denaturation and hydrolysis, difference between fibrous and globular proteins. That's 90% of exam questions. That's all for now. See you in the next video.